Hi everyone, my name is Patricia Parris. I'm a toxicologist at Pfizer and I'm also the rapporteur for the ICH Q3 Guideline. Firstly, I'd like to thank the Global Harmonisation Centre for the invitation to introduce to you today the ICH Guideline for Extractables and Leachables. And I'd also like to thank the IFPMA organisation for providing an expert to the working group and also for their support developing the guideline. I'm going to skip over this slide, but it's here, the legal notice from ICH. Just a quick, brief history really of the guideline. It's now, with great pleasure, tell you it's been signed off as a Step 2 draft document in August, and it's starting to be issued now across the regulatory members for public consultation. The draft guideline was developed based on the concept paper, which was back in July 2020. And so, yeah, it has been kind of a long road to get here. It's been hard work, but we are definitely making progress on our work plan, and our anticipated finalisation step 4 is going to be in June 2027. I have a few slides today really to introduce the guideline, some background to the Q3E guideline, some of the key principles that really are fundamental to how we've kind of developed the guideline, a high-level overview of the structure, scope of the guideline, and then a few slides going through the summary of each of the sections and the principles to the quality chemistry and safety sections of the document. And this is taken really directly from the concept paper, but the guideline is intended to minimise uncertainty to meet global regulatory expectations. It's to encourage the holistic science and risk-based E&L control strategy based on quality by design principles. And I will kind of repeat some of these things as we're going through because they really are the basis for the development of the guideline. We wanted to focus on critical aspects of E&L assessment and control to improve transparency and requirements for pharmaceutical products and also drug delivery device components. And there are a number of best practices and documents published in the literature about safety assessment, but this the ICH guideline allows us to kind of standardise the safety assessment and actually develop some qualification thresholds based on route of administration, indication, patient exposure, and also really emphasise that science and risk-based approach to the toxicological assessment as well. And we also have to be mindful of other ICH guidance, so complement and not contradict the existing quality guidelines and also ICH M7, and really, take this as an opportunity to align the diverse regional pharmacopoeias that are out there at the moment. So there's some of the key principles, and really this is extremely important, and it will be kind of, you'll hear it kind of being repeated throughout the slide deck, is this risk-based approach. And here we really relied on the risk management workflow that's described in ICH Q9. It's the fundamental framework to developing the E&L risk assessment, and it allows companies and then the health authorities to be reviewing scientifically justified extractable and leachables risk assessment. And of course, the primary purpose of all of this assessment and all of this work is to protect patient safety and ensure product quality through assessment and control of leachables in the drug product. And it really does require detailed material characterization and process understanding. And collaboration is a key feature of the guideline, ensuring you have good kind of connections and discussions between the chemistry, quality, analytical material experts with the toxicologists and safety assessment experts. I'm not going to dwell on this slide, but obviously we have defined we have provided a definition for extractables and leachables. So extractables are the chemical entities that are forced to come out under, stressed conditions, laboratory conditions that come out from materials, from the components, and leachables are the entities that migrate under the normal storage conditions of the drug product. Safety concern threshold is also an important concept that the PQRI group introduced, and that's the threshold below which a leachable would have an exposure so low as it would present negligible safety risk. There's a little asterisk here because there is an exception to this, which is for class 1 high concern leachables. And if the class 1 leachables are defined in the guideline, and if you think through your material characterization stage that you may have a material that could contain one of the class 1 leachables, then you should be measuring that because the PDE would be below that safety concern threshold. And how you define the safety concern threshold is it's by whichever is the lower for the drug product based on the route of administration and the treatment duration. You look at your ICH M7 thresholds of toxicological concern for mutagenicity, and then your ICH Q3E qualification thresholds for non-mutagenic endpoints. And you need to look across those and see whichever threshold is the lower, and that is your safety concern threshold. And of course, the SCT feeds into the AET calculation, the analytical evaluation threshold. And this isn't a control threshold, but it's a threshold corresponding to a concentration above which an extractable or leachable should be identified. And what we mean by that is elucidate the chemical structure, ideally confidently, if you can, and then it should be quantitated and reported to the toxicologist for the safety assessment. And the AET forms the basis of the extractable leachable risk assessment and the control strategy. So proper determination, so developing, calculating your AET is very important to the risk management process for extractables and leachables. And Q3E recommends establishing a study-specific AET, and that's done based on your maximum daily dose and, like I said, the appropriate SET for your drug product. It's a really critical step defining the AET in your whole ENL risk assessment. So here's a high-level view of the guideline organisation. So sections 1 and 2 include the scope and the introduction. 3 has the general principles, risk considerations, the documentation from a submission perspective, and that life cycle management once you might be making a change or something's going to change in the process or the product. And then we have several appendices that link to the different sections. So for section 3, there's appendix 1, and that includes some typical workflows on ENL risk assessment for both manufacturing components and also packaging components, and associated low risk scenarios with both of those types of materials. Section 4 has the chemicals testing and assessment, and an appendix 2 includes a summary of extractable, leachable, and simulated leachable studies. Section 5 is where we have the more details on the analytical evaluation threshold and things you need to consider when you're determining this. And there's some example calculations in Appendix 3 showing you how you integrate your maximum daily dose, or for intermittent dosing products or multi-day products, and how you consider that to define your AET. And lastly, section 6 is the safety assessment section. So there's a workflow here, details on classification of the leachables. So very similar to ICH Q3C, Q3E has class 1, class 2, and class 3 leachables to consider. Has some route-specific considerations, and there are 3 appendices associated with this section. 1 describing in detail the leachable classes. Appendix 5 describes methods for establishing exposure limits. This could either be using a margin of safety approach or has details on deriving a PDE, which aligns with the PDE calculation in Q3C. And Appendix 6 is where we include our PDEs that have been reviewed and consensus reached by the expert working group. So we have 2 class 1 PDEs. So these are substances that the qualification thresholds are not deemed to be low enough to control those class 1 compounds too. And the class 3 leachables, which I think are going to be more valuable, they're more the types of chemicals that we see all the time. These are your lower safety concern leachables. I think there's about 7 of those PDs, which was basically what we could get done and within the time frame we had. And those appear in Appendix 6 as well. So let's dig into the guideline, the scope. So organic leachables in new drug products are covered, and new drug products include cell and gene therapy products. drug device combination products that require marketing authorization to meet the definition of a pharmaceutical or biological product. The pharmaceutical part will be covered by Q3E. Obviously, the device part then becomes covered by the ISO guidelines. So those combination products are going to require looking at both guidelines to cover both aspects of your drug device product. Any life cycle management changes that relate to formulation, changes in the manufacturing, dosing, container closure system, and also storage of liquid or semi-solid drug substances. Another, it's important when you're defining the scope to say what isn't in scope. And so Q3 is not expected to be applied retrospectively to approved drug products. It's not expected to be applied to any extrinsic, extraneous, or foreign substances that result from product contamination. Herbal medicines aren't in scope. Clinical development products are not in scope. However, it can be useful to understand the principles of Q3E and start thinking about, how you're going to apply those when you get to your marketing, authorization stage. Excipients aren't covered, are radiopharmaceuticals. And I think the asterisk really is just to say that most principles in Q3 are applicable to inorganic leachables, but clearly the safety assessment of inorganic leachables is covered by ICH Q3D. So hopefully some of you will recognise the risk management process flow here, and this is from ICH Q9 and really just outlines high level this quality risk management process. And the expectation is this will be initiated with every product, and each will have its own risk assessment, risk control, and lifecycle management process. And the risk assessment step is composed of three aspects: hazard identification, risk analysis, and then an integrated risk evaluation. And so that kind of flow forms the basis of your material kind of characterization and understanding of your E&L potential leachable risk. And one other area which you'll hear me kind of say again and again, and is very important to the Q3E guideline, is prior knowledge. And so there's a whole section on prior knowledge that really allows you to, as a company and industry, to use existing knowledge that you have on your drug product, on your materials, to inform your E&L risk assessment. So whilst having a good understanding of information that you already have on that types of material or product, also understanding critical attributes for the manufacturing, packaging, and drug product components and the storage conditions. So having a very clear, holistic understanding of your product is important for being able to develop your risk management process. And as I mentioned before, in order to do this, it really does require increased collaboration between the analytical chemists and safety experts. You need that initial conversation to develop your AET, to understand what your safety concern threshold is. And then also, as you're then going through to understand, is there potential for any class 1s with your particular material there? Maybe you've only got class 3 compounds that you're seeing, and so you can use the guideline PDEs there. So having that close conversation with a toxicologist is really important to having a good, robust E&L risk assessment. And it's complicated. It was extremely challenging developing the guideline because it needs to be very case by case, because you have this risk matrix. There's a number of different considerations when you're beginning to think about your product, the conditions, the patient side. There's quality, pharmaceutical quality dimensions to consider, and also safety assessment dimensions. So some of these just listed out here, and each product will need to be considered in isolation and understand where each of these dimensions are, whether it's a low or a high risk situation. And we've actually depicted this as well as kind of having a description in ICH-Q3 with this risk matrix. And so it's just showing that, you need to consider each one of these and understand where your product sits on that risk matrix. And then that will inform your overall kind of risk assessment. And it is multifactorial, and it's complex, and it's meant that the guideline is quite high level. We are going to leverage the training materials to have some of that detail and give specific examples. But understanding this initial risk and the respected level and each of the factors is a critical part of the risk assessment process, and it may inform whether you want to use a particular manufacturing or packaging component, should you select that. But also, if you have selected it, it allows you to then develop your overall risk assessment and control strategy. And another key one of the biggest key principles is that your extractable and leachable strategy should be risk proportionate and justified. So that means that the expectation is that you'll be conducting leachables testing wherever possible, but there are situations where you have a significant amount of prior knowledge. If you've used the material for a very similar product for a number of years, and you have a good knowledge and understanding of what may leach out under particular conditions, then you can use that, and you can use that to justify why you may not need to do further leachables testing on a particular product. Using certainly for kind of oral products, using compliance with food contact or pharmacopoeial regulations, support your E&L risk assessment. So use the information that you have, but really document it very clearly why, if you're not proposing to conduct part of the testing paradigm, you need to justify that and explain why. And there are, to help with this, where you might be able to submit extractables data alone in lieu of leachables data. There are some low risk circumstances that are listed out in the guideline. So an abbreviated package may be submitted and may be accepted in these situations, but as you can imagine, that proper justification needs to be there. So we have a couple of typical workflows in the guideline. So one for the manufacturing components and one for the packaging component systems. And one theme that came out of the expert working group discussions were that for packaging components and systems, there is an overall lower risk, and that's recognised across industry and the health authorities. There's a shorter contact duration, a larger volume to surface ratio, and there are fewer extractables that are expected to be above the analytical evaluation threshold. And so this workflow gives you alternative options to use your supplier data, coupled with your prior knowledge. And if you have no extractables above the AET, then potentially you might not need to do leachables testing. And so that kind of works through that workflow. And as I said, there's some specific scenarios that are provided to help with that understanding of where is your risk acceptable and where is it unacceptable, and you'll need to conduct some leachables testing to have a better understanding of the potential patient exposure. And likewise, there's a workflow for the packaging component systems. It's obviously a little bit more complicated. You've got the product quality assessment as well as the safety assessment in there as well. But again, we were able to have, after a number of thorough discussions within the expert working group, we were able to define some examples where an abbreviated data package may be adequate. But I will say that from the health authority perspective, a comprehensive E&L data package is expected, and where you want to be able to provide and submit an abbreviated data package, that needs to be well justified and explained. So from the documentation and compliance piece, there is an expectation that E&L studies will be included with the rationale and methods, analytical performance and results, the safety assessment of any extractables or leachables above the AET, and there's a star here, or if you're in a situation where you've got manufacturing components and you're only submitting extractable data, that should be included. Your risk control strategy, demonstrating the adequacy of your proposed mitigation measures is important. And also that extractables to leachables correlation. And this is particularly important if you are in a situation where you might be observing a leachable and it wasn't there in your extractable study. So to be able to explain why this might be the case. And this is also can be very helpful when you're supporting your life cycle management changes, so changing a component, a filter, or such like, or even a stopper on the container closure system, demonstrating that correlation, and with the previous component and the current component is a very, very, very helpful and useful information, and is recommended to be documented and submitted. There's some examples of lifecycle management changes which could trigger further extractable, leachable evaluation. I don't think any of these will be a surprise to you, but if you have new information that could impact patient exposure or benefit risk, that needs to be considered. Changes to the drug product formulation, packaging component system, or manufacturing component systems. All of these changes need to have, at minimum, the thought process of gone through of, is there an impact to your potential leachable profile? A big section, I can't believe we've managed to kind of cram this big section onto one slide, is the chemical testing and assessment. So here there's a lot of detail around what prior knowledge is and what that means in terms of Q3E, and examples of information that you can leverage for your E&L risk assessment. You need to be defining and selecting and detailing how you've selected your component, and as industry, we're responsible for demonstrating an acceptable patient risk based on that risk matrix considerations. You might have extractable data. This could be semi-quantitative or quantitative, and in the majority of cases, a leachable study will be conducted for your drug product registration to understand the potential patient exposure in your normal storage conditions of your drug product. For drug products that have, kind of challenging formulations, there is the option to justify a simulated leachable study. So there's some details on that, on where that could apply or replace a leachable study when it isn't technically feasible to conduct it on the drug product. And as we mentioned, including that E&L correlation and understanding the source of the leachables, and if you do have any and they need mitigation measures, that is kind of described and explained in that correlation section. And I said before, defining that analytical evaluation threshold is a key critical step in your E&L assessment, and so that needs to be detailed and justified. It corresponds to a concentration above which the extractables or leachables should be identified, quantified, and reported for safety assessment. If you have semi-quantitative analytical methods, then you need to apply an appropriate uncertainty factor, and so that should be documented as well, what you're applying and why that's appropriate. And there are, again, multiple things that feed into defining that uncertainty factor. So it's the prior knowledge that you have, the materials of construction, the chemical structure of the compounds, and the availability of any reference standard to cover that range of response factors from your analytical method, and then also the limitations of the analytical method. So we'd absolutely relying on the expertise of the analytical chemists to help define the uncertainty factor when it's needed. I think that's the end of the quality chemistry section. We're going to switch tack now and go on to the safety assessment. And so this is really focused on the evaluation, or the toxicological evaluation, of potential leachables that are above the safety concern threshold. So any leachables that are below that safety concern threshold are considered to have no appreciable risk, and so no tox assessment is needed. As I mentioned at the start, the exception to this is class 1 leachables. So these are defined as high concern leachables. So if you've gone through your E&L risk assessment, your material characterization, and you have a material that could either have benzoapyrene, that's the possibility of the presence of benzoapyrene or bisphenol A, and the associated materials are listed out in the guideline, then these have a PDE that are below our qualification thresholds. And so you would need to analytically demonstrate that those class 1 leachables are below the PDE. And we've talked about how that safety concern threshold is product specific, and it's defined by the tox endpoints, that's mutagenicity or non-mutagenicity. It's defined by the route of administration and also the duration. And so this is where you really need that toxicologist kind of expertise to discuss with the analytical folks about the product, define what your appropriate SCT is for your study, for each study that you're conducting. And so I know there was a lot of, patience. Everyone knew that we were developing some new qualification thresholds, and here they are. So these are the ICH Q3E the current systemic and local toxicity thresholds. The local toxicity thresholds won't be changing. These are set, and you can see they're based on a concentration level, and they're split out by the routes of administration. The systemic toxicity thresholds are divided into oral and parenteral, dermal, transdermal, and inhalation. And we've also got the exposure durations, and we've aligned the exposure durations to ICH-M7. And so you have your TTCs, which offer the mutagenic endpoints, which are listed out here for each of the durations. And you have the QTs, which are your non-mutagenic thresholds that we have been deriving through all of the PDEs that we've been reviewing and developing within the ICH expert working group. And so these are our current numbers, and they will change slightly because the project is still reviewing, and the expert working groups are still reviewing the PDEs and reaching consensus on each of the PDEs. But how we set up the project is that we tried to send out the compounds that we thought were going to be kind of like the more toxic at the start. We have received all of the compounds back now, and we have reviewed the majority of the kind of more toxic ones, and we're now kind of in the non-toxic range, so we say. So I actually anticipate the QT numbers, if anything, slightly increasing, but this is where we are now. And I think it's interesting to note that in terms of the oral QT, that less than 1 month is kind of around that 120 micrograms for the mutagenic TTC, which is it's nice. That's kind of aligning, and then you can see obviously the parenterals are slightly lower because we have that additional adjustment factor for bioavailability, because most of the studies we've been looking at and developing PDEs from have been by the oral route, and so we need to add in a factor to account for that route extrapolation. But yeah, these are where the numbers are now, and we have very much focused on developing oral and parenteral qualification thresholds. There is an understanding that dermal thresholds and looking more into the inhalation thresholds have been kind of pushed to the step 2, and so we've got these kind of temporary thresholds in here based on the parenteral QT, because that's expected to be a worst case. We will be looking into developing, further route specific QTs in the next revisions of the guideline. There's also a safety assessment workflow that takes you through how you look at your data in terms of the different mutagenic and non-mutagenic thresholds that are out there, and where you would then need to, if you've identified a potential safety risk, kind of loop back to the analytical folks and start deciding on defining and designing your mitigation strategy. So a safety assessment is going to be conducted for all observed class 1 leachables. So these are leachables of higher concern. For class 2 leachables that are detected above the relevant safety concern threshold. Class 3 leachables when they're present above 1 milligram per day. So this is very much a similar approach that Q3C took, where we've developed PDEs for a number of common, relatively non-toxic chemicals, and we've set a 1 milligram kind of threshold. Each of the substances that have a PDE are actually a PDE above 1 milligram, but we felt from a quality perspective 1 milligram should cover most product situations. And so if you're below 1 milligram with your class 3 leachable, you don't have to do anything. If your potential patient exposure is above 1 milligram, then you can go to the supporting documentation, which has the individual PDEs for each of the class 3 substances, and use that for your toxicological assessment. And your safety assessment should be demonstrating the acceptability of the anticipated patient exposure levels by considering these different endpoints. They've got to consider mutagenicity, non-mutagenicity, general systemic effects, and also if it's a particular route of administration where this is important, local toxicity effects. Here's some more details about the potency classes, and this is included in Appendix 4. So these class 1 are leachables of higher concern, and these are aligned with the class 1 ICH M7 class 1 with an acceptable intake below 1.5. So these would be your cohort of concern mutagenic compounds. These should be avoided, or a compound specific acceptable exposure level should be established. And our guideline currently includes benzoapyrene, which is associated with carbon black. So if you know you have carbon black in your material, then you should be looking for benzoapyrene to demonstrate it's below its PDE, and bisphenol A. And the materials that it's associated with are also listed. And again, that's B where you would look for those. And not surprisingly, we don't have many class 1 compounds. We are performing material, well defining our material characterization, and so we're not expecting to see compounds of high concern. I think it's just comforting that the analysis that we did confirms that there are not a huge raft of potentially highly toxic substances that we see as leachables. I think most leachables that we see will fall into this class 2. So this is the default classification where the mutagenicity TTC and systemic tox QT are considered to be sufficiently patient protective. It's ICH-M7 class 1s with an acceptable intake above 1.5, and also ICH-M7 class 2 or 3 impurities. And it's obviously, but I guess it's good to lay it out here, non-mutagenic leachables that don't fall into class 1 or class 3. And the class 3 are our leachables that are expected to have a relatively low potency for systemic tox. Their chronic parenteral PDEs are going to be above 1 milligram, and they are considered qualified up to 1 milligram, independent of the route. Or you can, if you're above 1 milligram, then you can look to the compound-specific PDE. And the guideline currently includes the chemicals that are listed out here, and hopefully you'll see these will be, leachables that you're familiar assessing. They're common leachables. We basically picked these because we know they're seen relatively often, and this was the most number we could get in within our time frame, to meet our current time frame. So we do have a list of additional chemicals and PDEs that we will be increasing this class 3 group as further revisions of the guideline come on, but this is the ones that we were able to get done, and hopefully the most impactful for revision 1. The toxicological kind of risk assessment principles are hopefully for the toxicologist of view. I felt this section was fairly straightforward. I don't think there was anything very controversial. I think the big thing about the safety section was developing those harmonized qualification threshold that everyone kind of could agree on and feel confident and believe in those qualification thresholds. They're scientifically justified qualification thresholds. I think the safety assessment principles are hopefully how most toxicologists are conducting their leachable assessments. So you can either define a compound-specific PDE, which would be for chronic exposure, or it's an acceptable exposure level if it's a less than lifetime exposure. And that's aligned with the Q3C and Q3D calculation, or you demonstrate a large ratio between your justified no effect level and your patient exposure. The guideline encourages in silico analysis and also using read across approaches. So really try and use all the approaches that you can before you need to do an in vivo study. That's kind of like really the ethos of the guideline. So all approaches should be considered, and your last resort will be conducting an in vivo study. So we're getting to the end now. I think there's a couple of slides I have. So the timeline, as we mentioned, we're in step 2, AB public consultation. EMA comment period ends in mid-December. I don't believe the FDA has come out yet, but I think we're looking at hopefully having the consultation periods closing and the comments back in the new year. So kind of January, February time, 2026. We actually have a meeting. Our next face-to-face meeting is scheduled for March, and this is a safety focus meeting to really finalise the threshold project, those PDs and the qualification thresholds. And then, as I said, step 3 sign-off and step 4 adoption of the guideline is currently scheduled for June 2027. Here is the awesome expert working group. They're such a fantastic team, so enthusiastic. They've put up with me putting weekly, meetings in all over the shop. They've really stepped up and had some really excellent scientific discussions. This document is the output of us reaching a consensus, a harmonized position across industry and regulators. And we're very proud of the document and are really pleased that it's out there, and looking forward to receiving everyone's comments on the guideline. Thank you very much. I'm happy to take any questions now, and thank you very much for listening.